Hello everybody, this talk is about our simulation of cellular automata in Oritatami model. This is a joint work with Nicolas, Schabanel, Shinnosuke, Seki and Yuki Ubukata. In molecular assembly, the sequences of nucleotides are folding into shapes. This is an active area of research, gathering both theoretical and experimental results. Today, with DNA folding, we can assemble complex 2- and 3-dimensional shapes in nanoscale, like you can see this picture on the top. It's impressive. But moreover, we can perform computations with DNA. There are several theoretical models for the DNA assembly, and the most known one is called the abstract tile assembly model. And it was proved to be Turing universal and, moreover, intrinsically universal. That means that it can simulate any of its kind. Among experimental results, we have this folding of Sierpinski triangles and a set of reprogrammable DNA tiles able to execute several 6-bit algorithms. But DNA folding has its disadvantages. For instance, DNA molecules are very rigid and they require high temperatures to be assembled. That's why we're interested in the RNA folding. RNA self-assembly is like a greedy algorithm because the molecule is folding directly while being synthesized. More precisely, it happens like this. We have an artificial DNA strand, gray here, and the RNA polymerase is walking on this strand, producing its copy called transcript, blue, this one. And the transcript is folding upon itself immediately after being produced. And that's why this folding is called cotranscriptional. The experimental results of Gary, Rothman, and Anderson in 2014 showed this folding to be quite promising. In contrast to the DNA folding, the RNA folding is happening locally around the polymerase, where the transcript is flexible and creates the connections easily, and it doesn't require high temperatures. So the Oritatami model is a simplified model of this folding, introduced only 5 years ago. Let me give you the theoretical definition of the Oritatami model. First of all, we live in the triangular lattice. The nucleotides are represented by the set of bit types, defined alphabet. A chain of bits is called a transcript. And when we put our transcript in our triangular world, it's called a configuration. Now, the hydrogen attractions between bits is represented by a symmetric relation called an attraction rule. When two attracted bits are adjacent in a configuration, they create a bond. The bonds are marked by red dotted lines here. The number of bonds in the configuration is denoted by H. The more bonds a configuration has, the less energy it has, and the more stable it is. So now, a duly produced flexible part of a transcript is called a nascent part, and its length is denoted by delta and is called a delay. Consider a configuration. A prolongation of this configuration by delta bits is called a delta elongation. And the set of all delta elongations is denoted as shown here. Now we will define a step of folding of Oritatami. So we have an Oritatami system. It consists of the set of bits, the transcript, the attraction rule, and the delay. Given the configuration C, we will now find its successor in our dynamics. For this, we consider all the delta elongations of C maximizing the number of bonds. For instance, this one has only one bond, as well as this one. But this, and this, they have 3 bonds which maximize the number of bonds. And so if all of them agree on the position of the first nascent bit, then the successor is just C plus B, this first nascent bit. So the formal definition is given here on the slide. So now we're ready to define the full dynamics of the whole process of folding. So we have an Oritatami system, and we start from the initial configuration of the first bits of the transcript, C0. It's called the seed. So in this example, we have the first 3 bits of the transcript. And now, at each step of folding, we find the successor of the previous configuration. For this, we consider all the delta elongations, choose the ones that maximize the number of bonds, like here it's only this one, and we fix the first nascent bit, and we get C1. And with the same actions, we get C2, C3, C4, and now we have no more bits. The transcript is over. So this is the last configuration, and this is called terminal configuration. So now we have the full theoretical definition of the Oritatami model, and I will show you what's known about it. First of all, one of the goals in this domain is to build shapes. So we know that in Oritatami we can never fold infinite shapes with a finite cut. But for any other connected shape, it can be upscaled and folded using only 19 bit types with delay 1. The other goal is to build smart objects to make computations. So with Oritatami, we can simulate a binary counter, and moreover, we can simulate any tax system, which makes this model Turing complete. Besides that, we know that the rule existence is NP-hard. As a result, like the Sierpinski triangles for DNA, we can build a finite part of the highway dragon fractal with Oritatami. And now the work in progress is to create a system building an uncomputable shape. So right now, we will talk about our simulation of a cellular automaton. To define a cellular automaton, we first need to choose the finite set of states Q. Here it's 0 white and 1 orange. Then we take a neighborhood of a cell in a one-dimensional discrete line. Here it will be the cell itself and its adjacent cells. And finally, we choose a transition rule, which takes a neighborhood of a cell and returns the new state of this cell. So now, to run a cellular automaton, we choose the initial configuration like this one, and at each step of computation, we apply our transition rule to each cell in parallel, like this. And so we get the next configuration, and again, and again, and again. And finally, we get the whole time-space diagram of a cellular automaton. Here is a discrete version of Sierpinski triangles. A very important thing to know is that cellular automata are Turing complete. Now we will simplify our task, and instead of folding cellular automaton, we will fold 2 into-out gates Rhombus automaton, which has 2 input neighbors and 2 output neighbors. So to the left, you see one step of computation of a cellular automaton of radius r. And here at the center, you see how to simulate this step with r+1 steps of our Rhombus automaton. So now our aim is to fold a space-time diagram of a given Rhombus automaton with Q states. We do it folding cells one by one in a snake-like way, like this. Our transcript consists of the repetitions of the same segment corresponding to one cell of the diagram. This segment is divided into 6 modules. So the first Init model will be discussed a bit later. The red scaffold model is building a skeleton of our cell. The third blue model is reading states of the neighbors. The orange module plays the role of the transition rule and prepares for writing. The synchronization model will be discussed a bit later. And the last one, the writing model, is transmitting the output to the borders. So now we will get into some details. First of all, after the scaffold is created, we start reading. The reading module is folding into zigzag normally, but the neighbors of the cell put two magnets on the side of the cell corresponding to the input of the cell. The input is X and Y. And so in front of the magnets, the reading module flattens and goes straight, pushing the molecule forward and creating a shift of Qx plus Y. And so during the next orange module, the shift is still present, and it plays a role of transition table, and the orange module puts two markers, many markers actually, to prepare for writing. We'll use them later. Then the synchronization module absorbs the shifts because the writing part should happen at a very precise point of the transcript, independent on the input X and Y. And finally, during writing, we use the markers we placed during the orange module to put our proper magnets and transmit the output. Now let's discuss the synchronization module. So its aim is, if there is no shift, to go straight, but if there was a shift created, it should fold upon itself to absorb it. A bit more details. So on the top, you see the scaffold part already folded. It's highlighted by red. It consists of blue and pink segments. So the synchronization module, going from right to the left, it consists of the same blue and pink segments. And so when there is no shift, all blue segments are in front of blue. And so the synchronization module goes straight. But when there was some shift created, then some blue bits are in front of the pink segments. And in this case, the synchronization module goes in a zigzag way, absorbing 1/2 of the shift each time. And so if we had a shift of length n in log n steps, it will be absorbed completely. So now you remember that during folding, we're going in this snake-like way. And so that means that in yellow cells, we need to turn and change the direction. And the init module takes care of this. So in normal cells, when we go straight, it just falls upon itself in the beginning of the cell in this manner. But when we have no neighbor, we should turn. And so in this case, it goes straight and creates a wall, and then it turns. Notice that this is the same segment of the transcript behaving differently in different contexts. So I gave you the ideas of our construction, but I hid all the rigorous computations necessary to align everything together. So you can look at the picture and have an idea of how complex it was. Besides that, another challenge was to verify that everything works correctly. And so to see that the whole structure works as it's expected, we needed just to check it for all local configurations, because the Oritatami system is folding locally. And so to conclude, we designed an Oritatami system simulating any one-dimensional serial automaton, and it features very simple modules which are easy to understand and to use, in contrast to the previously constructed tax system. And besides that, this module can be reused for the further project, like the synchronization module, magnets, and init, and we're already using them in our system, making a non-decidable shape. So the next step would be to implement loops and some other stuff to design a Oritatami compiler, and because we're moving between the experimental and theoretical domains. We're very interested to implement some simplified tools of our construction, like the synchronization model, in the wet lab. So that's all. Thank you for your attention.